It's a really pleasure to be here this evening to talk to you about our work on imaging and preclinical models, and touch a little bit as well on our adoption of the Bruker compact MR scanner. So I'll be showing some work today that ranges quite broadly from the experience we've had in using imaging to look at cancer as well as other diseases, and showing some results from a number of developments internally in terms of technology, but also different companies that we've worked with exploring some novel biological questions and bringing imaging in to answering some of those questions. I think it's pretty clear to everybody, next generation cancer intervention is going to come from a variety of sources. Improving the quality of current interventions, understanding what we're doing, whether we're doing it effectively, enabling combination therapeutics, which is something that's hard to explore and understand how to apply optimally in patient populations. And preclinical research is going to really help us understand the combinations. And also looking at adaptive or stratified or personalized medicine contexts, understanding the fundamental underlying processes that are at play. And it's clear that imaging will play a central role in improving current therapies as well as advancing new ones. And I'm sure many of you have seen these really nice figures just published in Padani in 2010, just highlighting how you can image the various hallmarks of cancer as different probes are becoming available to try to let us explore at different depths the processes in an in vivo context. So imaging allows us to look at longitudinal, multimodal, multi-scale characterization. I'll show in my talk some examples where you can really pull modalities completely from microscopy down to bulk imaging and try to link these two. Also, precise and accurate targeting of the intervention to really know what we're doing. Asking questions like, can we use imaging to understand where the drugs go, for example? Are we really delivering what we think we are? And can be employed to assure consistency of neoplasm development. We'll take a neoplasm, we'll apply a therapeutic. That neoplasm is a wide range of phases of development. Having more consistency in this is a very powerful tool, and imaging can help. There's a great editorial by Nurse, of course, Nobel laureate, and highlighting that the whole complexity of the biological system is really going to be supported and enabled by integrating imaging into our observations. And I think this is really some of the thinking that's been driving the adoption of imaging in preclinical models. This is a great slide I just borrowed from a friend of mine, Paula Foster, from the Robarts Research Institute in London, Ontario. And she's just actually looking at not cancer, but in fact spinal cord repair. And she just showed this in her talk the other day, and I thought I would share it with you. And I think it highlights one of the elements that imaging in cancer research can be quite useful for. And she's just looking at where stem cells go. It's in a cord regeneration model. She put some iron oxide nanoparticles in some cells, and they were injecting them to see if they could return function of damaged cord in a mouse model. And she was applying this with another lab in the institution. And this is the first time they ever imaged where these cells were going. And she showed very nicely, if you put the cells in the spinal cord, you can actually see them migrate along the cord, and they would demonstrate different degrees of recovery. But 4 of the 10 animals they studied had this happen. It's a little hard to see, but in fact, the injected cells never got into the cord. They just assumed it was a variation in response in that population, but in fact, they didn't have the evidence to show that it actually got into the cord. And I think imaging in the context of intervention is a very powerful tool to make our research more precise. So science and tools are closely linked. So I'm going to show you some tools that have really brought forward some interesting science in combination with imaging. So I had the fortune back in 2005 in leading a large grant to put together a very comprehensive preclinical imaging facility. And optical systems, ultrasound systems, CT. We bought a Bruker 17 magnet that has been working very hard for several years. Dedicated animal housing resources, SPECT, PET, and micro-CT. I'm going to draw from these over the rest of my talk and just show you some examples how we've used these imaging resources. And most recently, a couple years ago, we got the Aspect 1T magnet, and that's recently been switched up to the Bruker Icon, and we're integrating that as a valuable tool that complements the 7T. We run this facility as a core, but it's also my lab at the same time, which is quite fun on some occasions, quite stressful on other occasions, grant cycles and the like, because there's people to pay. But Justin runs the facility, Deb heads up all the preclinical studies, and then we have different scientists and staff that work on different modalities. Warren Foltz takes care of some of the MR issues, and Doug Vines runs the PET. And so this team operates. I thought I would just present that in case anybody was interested in this kind of model. In fact, it's a dedicated operations team. It's called STARR. I'm not going to explain the acronym. Maybe you can stay afterwards and buy me a beer, and I'll tell you what that means. We do the imaging and agent development, veterinarian support, image analysis, and now tissue processing, an end-to-end approach. Dedicated facilities for all the elements. And since around 2007, when we opened up, we've had over 290 registered studies, different preclinical research studies, and 70 investigators. And if you're interested more in the facility itself, it's at STARR.com. We bring the elements together, PET, CT, MR, and now with the ICON as well as other modalities, image registration, image analysis software, and results. These are kind of pieces you need to pull this off. It's good to get some images, but when you want to run it over and over again and have a stable assay, it's really useful to think about these tools. And I think there are some good tools emerging now in the market. We start to think about how do you pipeline, like the question earlier today, how many animals can you put through? This becomes quite key, and if you have drug available in the morning that's going to taper out over the course of the day, how do you stack up multimodal imaging studies? Because the multimodal element is becoming quite key, not just look at the anatomy, but look at the underlying function. And tools that let you move the animal readily from one setting to the next and be efficient in this activity. So just to give you a bit of a feel, as an example of a novel technetium-based hypoxia marker, where you can get the images on a SPECT, overlay them on CT, but you'd like to be able to confirm that autoradiographically, and then link that back to tumor H&E sections, possibly with EF5, confirming the distribution of the hypoxia. Because the image is nice, but what does it mean in terms of actual biodistribution? So rather than talk about a bunch of hardware, I'm going to talk to you about just some experience we've had using these systems and give you some insight into some of the problem sets and some of the challenges. This is an interesting project. We call it the Microenvironment Imaging Pipeline Project. And the idea here was to think that imaging is a phenotyping tool. And the questions that came up, we were taking tissues out of patients and putting them in a biobank, and then we're transfecting those, of course, into mice and growing patient-derived xenografts. This is a very topical activity right now. In our institution, about 1,000 banked and functioning patient-derived xenografts from all parts of the body. And this one in particular was from colon cancer. It was a project funded by Pfizer and the OICR. And we went through the process of having animals, each running a specific patient-derived xenografts, and performing imaging studies. This ran over about 4 years now. Dedicated FDG micro-PET, looking at micro-CT to look at the degree of perfusion in these tumors. The tumors are then sent for staining, looking at hypoxia characterization there. We also go on and do large whole mount visualization, assess metabolic bioluminescence imaging techniques to look at these tissues that have been snap frozen and look at specific characteristics of the metabolic components in the tissue. And then those cells are also banked, and of course they're going to go then to flow cytometry, staining and analysis. And then of course linking this all back together and analyzing and understanding, did that specific tissue removed from that patient have a consistent imaging phenotype in the subsequent placement in tissues in mice? So do we actually have an immortalized disease that we could passage through time? It's a very complex problem. In fact, the passaging process seems to also impact the characteristics of this. And so we're in the process of doing this and collecting this data through the infrastructure. So far, we've phenotyped about 22 models and 105 mice. These are different numbers of how many mice have had FDG or DCE and the various assessments. And then another 8 of those 22 models have gone against specific drug interventions to test and evaluate. And I'm not going to dwell too much on some of the results, but just to give you a little hint of what we think will come out of these kinds of studies. But across the bottom, this is colon cancer, and these are specific patient-derived xenografts. And what we're starting to see, this is just looking at FDG distribution, and this is at a specific number of passages for these patient-derived xenografts. But in fact, there appears to be clustering. In other words, the tissue that came out of one individual human appears to have specific imaging phenotypes that propagate through time. If they didn't vary from patient to patient, they would be consistent across the board. But I mean, you have to be cautious. I mean, there's so many things at work here. You'd want to be able to compare to other metrics and make sure the data set's consistent. So the observation that came out of this study was interesting. Xenografts seem to show consistency in percent injected dose per gram FDG uptake, and within the same model versus across models. So maybe there is an imaging phenotype that we'll have to understand and characterize. Interesting to look now how that links to the genetics of this disease. But also we looked at the lactate characteristics, and we didn't run as many animals, but as many of the models, but we ran a number of them. And then we could ask the question, how does FDG uptake and lactate accumulation, how do they correlate? Because you would think there would be some relationship, at least at a simple level, between those two. And in fact, we were able to demonstrate that the lactate concentration uptake and the FDG consumption rates were correlated. So now we're starting to see that the data set has some consistency. And this is just a first beginning in trying to understand how we can link these various metrics. I'll give you another example of using imaging in the context of novel cancer development. This is a collaboration with a company called Merrimack out of Boston that we've been having a lot of fun with. Liposomal drug delivery systems we know have been perceived to be very successful in the past and then didn't work as well as we thought they would, and now they're coming back again. There's always been some questions about why that is, and there's many hypotheses. A perfect example of image-guided drug delivery analysis was published by Harrington, the reference has gone off here, back in 2001, where they labeled liposomes with indium and then looked at the differential uptake in a number of patients. And these are 17 patients, a variety of different types of tumors and different specific diseases, but then just looked at the percent injected dose per kilogram. And I'll just pull this up, and you can see these are all three: breast, ductal carcinoma, T3, T4. And the percent injected dose per kilogram varied by a factor of 3 in these patients. So the question that comes to mind is, why is that variation? But wouldn't you expect one patient to perform differently from the other depending on the drug delivered? Now this is vehicle, not drug, but it's reasonable to ask, if you don't get the vehicle there, will you get the drug there? So there's a company called Merrimack, and they have a novel therapeutic agent called MM302. And it's specifically a liposomal system. It's targeted to, not because it actually gets more to the tumor, but because it gets internalized better within the cells, is from their evidence. And they asked the question, could we take an imaging agent and build an imaging agent that would first determine how well the drug gets there? And if we knew how well the drug gets there, maybe we could pre-select which one of those patients. Some are radiotherapy. In radiotherapy, we know the dose to within 5% to a target. That's standard of practice. And so I found this is very interesting. Of course, in the context of drug delivery, you'd like to know that the agent is actually getting there. So we ran this study within STARR, and I thought it was quite interesting, and in fact used the aspect magnet to look at various response. And so the study was pretty simple. Here we have our imaging agent, which is labeled with copper-64, and we assess first the distribution of that within the animal, and then we apply treatment, and using MR, which is quite relatively simple to do, as demonstrated in the previous talk, look at the volume reductions through multiple courses and beyond for the specific therapeutic. And you can see here the procedure is pretty straightforward, the PET imaging and CT imaging to look at the uptake of the imaging marker, and then subsequent treatment and monitoring with MR. And this is with collaboration with Helen Lee and Jinzi Zhang from within our facility, and Helen Lee from within Merrimack. And so we just started to ask the questions, what's the percent tumor change week 7 relative to week 1? You can see you had wide variations in the percent tumor change. Some of them grew and some of them shrunk. And we tested that against the magnitude of the uptake of the image-based, the imaging agent, which was sort of a test for drug delivery. And there's a really strong correlation. The greater the uptake of the drug at the beginning of therapy correlated very well with the reduction in tumor volume change. And of course, this model is attractive from a personalization point of view. If you could produce this imaging and therapeutic pair, you could pre-select those patients which demonstrated significant uptake to go on to the trial and and enhance the likelihood of success for those specific patients. We also looked at some issues around tumor deposition variability between different types of tumors. And of course, those were sub-Q or in a fat pad had slightly different variations, but insignificant in terms of their total number. One of the interesting elements in terms of the treatment of cancer, of course, radiotherapy is a very large part of that. And we started to work in the imaging context and realized, how do we mimic the radiation therapy delivery elements in the preclinical setting? Traditional radiation therapy models are not specifically targeted as we have in the clinical context. Now, this is an ICRU report from 1962, and I thought I would just pull some nice figures out of sort of radiobiology type of experiments and the kinds of irradiators that were used and still are used for different kinds of studies, but they didn't really have the ability to conform the radiation dose within the subject. Furthermore, we know now that drugs in combination with more aggressive combined radiation in combination with more aggressive combined therapeutics can produce unacceptable toxicities. So if you really want to understand the combination of radiotherapy and a drug, you need to be more thoughtful about the volume that is being irradiated. So this is a conflict of interest. We worked with a company, Precision X-ray, actually Richard Hill and I, Richard's a radiobiologist, realized we needed to get rid of our old X-ray system, and we designed this new one. And we worked with a guy named Pete Rollins at Precision X-ray, and it worked so well that somebody else asked us for one. And so we went in the process. Now we've made 12 of these machines that allow us to do CT and targeted radiation. And it's been very interesting to see how this new tool has brought about new biology, and now we're linking this new tool with the work on the ICON. And this just gives you a bit of a feeling for the system. It's very much a mimic of image-guided radiation therapy systems. It takes a CT scan of the mouse by rotating around the mouse. The mouse is positioned on this little table, and imagers are then produced that allow us to direct the mouse then such that the mouse's target is at the isocenter of all those beams that would rotate. And I'll show that in a little picture here. Images come up. You can kind of say, I want to treat this target, and then you can shift the mouse to locate the radiation. And this just sort of illustrates the kind of conformality one can get in the steepness of the dose gradient. So this is now pushing all kinds of interesting questions forward about how we can actually mimic, as we heard earlier from the first speaker, this sort of mouse hospital model, including integrating radiation, which is a therapeutic, but it's also a very interesting biological stressor. And you can use that to explore a variety of interesting biology. And of course, I mean, if you build this, and people who know radiotherapy, you have treatment planning systems to design this. So now groups have produced treatment planning systems so that you can actually design these beams. And this is a Monte Carlo-based system that shows how you can predict and calculate the dose from these kinds of devices. And now companies are actually getting involved. A human treatment planning company called Research Labs out of Stockholm has sort of worked with us and brought their entire suite to bear on this, so we can actually think about exactly where the radiation dose is going, and realizing that in these kinds of energies, a lot of dose goes into bone, and how can we control and place that radiation dose. So with a tool like this, you can really start to think about some interesting things. Now, I wanted to show this video because my student, James Stewart, I asked him, how complicated of a dose pattern could you make? Because we could really explore some interesting biology. Maybe we could look at how stem cells migrate if we produce complex patterns of irradiation. So he's an interesting guy. So he's there, he took an image, now he's adjusted, and you're gonna see, it's kind of a neat system because this film, you'll see, he's putting a collimator, it's kind of sped up. This film is called radiochromic film. It changes color when you irradiate it. So I said, just put a camera on the table and we can watch. And it takes a little bit of time, but he demonstrates the system's ability to paint the radiation dose. And you can see that it's quite small. I mean, it's a little bit disappointed at his handling of this part of the project here. But anyway, you can kind of get a feel for so it's a really interesting combination. The ability to visualize where these things go and to control their placement is opening up, I think, quite an interesting domain. So we had, that's a little, I don't know if you know Richard Hill. We also did a picture of Richard Hill with the irradiator. We thought that was kind of a cute project. But anyway. So with a tool like this, we can really try to do clinical trials that could be relevant to clinical context. And we had a radiation oncologist named Carolyn Chung, and she published this paper just actually last month in the Red Journal, where she wanted to understand, are there any image-based markers of the combination of sunitinib and radiation therapy? or actually any other therapeutic in radiation therapy. And so she literally ran a very sort of complex clinical trial within the STARR environment. There's a U87 glioma cell model in non-skid mice. And after the tumors were placed, and we were confirmed that they were there by MR and knew where they were and their size, we could actually route these mice onto multiple arms. And we could look at survival analysis, but in a subset, we could go a lot further. We could actually image some of them and look at the distribution and growth of the disease. And in addition, we could run a number of imaging studies, for example, serial multiparametric MR, looking at perfusion and response, and ADC to see what kind of response we're getting in terms of tissue. And so just to illustrate this, so you have mice you can actually visualize as demonstrated in the previous talk, and this is the implanted tumor, very well-defined and clearly localized, consistent in their size. So we're introducing a very consistent size of tumor, well located, into a controlled combination of radiation therapy and sunitinib in a comparative trial. The sizes are pretty uniform. The variation in size is only a factor of 2 in volume, which is pretty incredible in terms of the control. And we can be sure that it's where we think it is. This is, for example, a typical location. This would have been kind of questionable because it's too far lateral in the control of these studies. It's the dark forces entering the room. And so, of course, if you have the MR image, you can fuse that to the CT on the irradiator, and you can deliver then conformal radiation dose to that volume. You're not delivering a large dose to, for example, the swallowing apparatus, which would influence the ability of the mouse to survive. So we get a nice, clean experiment. And then you can look at survival in these animals. 36 mice were followed to survival. Those that had the placebo, those that had radiation alone, those that had sunitinib, and those that had the two in combination. And it's possible to sort of now ask the question, what was the optimal? But what Carolyn Chung wanted to do is she wanted to ask questions about, are there any early response metrics that we could pull out of imaging to tell us which of these mice are going to respond differentially? Because that's very important for patients who have, of course, this kind of disease to know whether or not continuing on a therapy is worthwhile. And so we had analyzed the apparent diffusion coefficient. The apparent diffusion coefficient is something that you can measure with MR. Basically describes the ability of protons in water to move in their environments. And if it's a low coefficient, then water is well bound. If it's high, the water can move. And so cellularity would suggest low diffusion coefficient. Loss in cellularity would suggest an increase in a diffusion coefficient. So here you have a baseline image. You can beautifully see the lesion. And we're looking within the region of interest of the enhanced on the MR. So now looking only in the region on the MR where the enhancement exists to see whether or not we're seeing a change in the ADC. Not looking over the whole brain, not looking over half the brain, but only where we know the disease. So we're looking at the response of the disease-carrying portion of the brain, not the surrounding normal tissue. So it's a very explicit and careful methodology. And we can look at the percent ADC in the tumor compared to the ADC on the contralateral side of the brain as a function of time. The radiation therapy shows a much more rapid change and greater change in apparent diffusion coefficient that's occurring in the tumor compared to those that were control or with sunitinib alone, but in combination you got this intermediate point. So Carolyn asked the question, in day 3, is there any predictive capacity? It would be nice to know after day 3 whether we should continue or not. And so this is just using the MR signal, and she pulled them out, and she was able to demonstrate that the relative change in ADC between day 0 and day 3 was predictive for a growth rate differential. In other words, those that did not have very much a change early on ended up with a more aggressive growth rate, and those that had a response were reduced. Now, she just compared the cohorts, the 4 cohorts, but we have all the data. We have every image for every mouse. So she said, let's pull them out of the cohorts and see how the individual mice results predict. In fact, here she shows for the individual mice, because she has the data for them, there's no need to group them. You can look at them individually. And this is the power of these kinds of tools in the clinical context. You could see that, of course, that the change in ADC on day 3 predicted very strongly for tumor growth rate. It's remarkable capacity to have the specific information. Otherwise, if you don't have the information, you have to group these into cohorts. There's no way to pull this out. So another interesting project we had with radiotherapy and was to integrate optical monitoring. I thought this was really interesting, and we'll pull this back into the MR at the end. Ralph DaCosta was a physicist who liked to do photonics work, and I tried to convince Ralph to do radiation work. So I built the machine, and he's used the machine to use optics to look at how tissues respond. But Ralph is a photonics guy, and they like to look at things up close. I mean, they're like this, microscope thinkers. And Ralph thought, well, I'm gonna take a window chamber model, and I'm gonna look this beautiful vasculature in the window chamber model, and then this irradiator, you say you can put a 0.5 millimeter beam on it. I said, yes, I can put a 0.5 millimeter beam on this, Ralph. And so he said, well, let's irradiate in a very controlled, very directed way tumors in the window chamber model. And so I said, well, okay, let's make sure we know where the dose is. So we took that radiochromic film that I showed you a little earlier. This is a little different color. We actually put it in the window chamber, and we put it there when we irradiate. So we had absolute confirmation of what dose was delivered and exactly where it was going. In fact, you could take a profile of the dose right across here and say, this is the dose that was delivered, and this is exactly where it was delivered, and this is exactly where it was delivered relative to this microscopy. Because this window chamber can then be put on a microscope, and you can do confocal microscopy and a variety of other things to now look at what's going on in these window chambers. So Ralph published this just last year using in vivo optical imaging of tumor and microvascular response to ionizing radiation. And he looked at white light images, the application of the controlled irradiation, confocal microscopy, as well as OCT, which is like an optical version of Doppler ultrasound, okay? So think about like a really high-res ultrasound Doppler image. And this is a really nice demonstration. So we took an ME180 tumor model, and these are DS-red labeled. So these are tumor cells, cervix cancer tumor cells, the patient died from a long time ago, but they're marked red. And he injected them in the window chamber, and you get all this nice vasculature, and you put Ph.D. dextran. You can see the vessels all light up in this green from the Ph.D. labeling. And you can see then this is pre-radiotherapy, and we irradiated the tumor. And you can see over the course of time how the vasculature recedes. In fact, it's a little odd. The tumor seems to get sort of bigger, and then the vasculature recedes almost completely at day 10, 14, and then day 20 it's starting to come back in. And this shows the flow characteristics that are also knocking down over that time characteristic. You can see this in specific detail. Now Ralph's gone a little further. He's now saying, okay, I can put a window chamber in a skin, but where else can I put it? Now I'm just going to show you two other window chamber examples, but this is we're going to run out of time. So this one specifically was to put a pancreas window chamber. So this is a window chamber now attached, so you can see the pancreas. And then you can do a lot of things. You can look at optical with the Ph.D. and the DS-red labeled. You can also look at photoacoustic to look at blood flow and ultrasound. And you can get, of course, 3D ultrasound. But then also you can lay MR onto this and visualize, because we're going to irradiate ultimately onto this coordinate system. We want the MR to be able to characterize things like magnetization transfer, which is, of course, something of interest in pancreas. And this just shows now an image of the window chamber. And the window, it's an MR-compatible window chamber. You notice the metal's all gone from around it. And we have our orienting slice. And now we're marching through that window chamber, and you're seeing the pancreatic tumor lying just below. So we've got literally all, now I guess all I need to do, of course, is apply radiation therapy, because we can then take that and position it, see the fluoroscopy, do the cone beam CT with the window chamber, and place the radiation dose and confirm it with our radiographic technique. And then we can look and see, using how photoacoustics, which is a really interesting imaging modality, but allows you to look at the amount of oxygen carried in blood, both pre-treatment and 1 day post-radiation therapy. Now this is early work, which is starting to get to the point where we're getting close to publishing it, but just to demonstrate how you can pull these different techniques together. So now we're moving forward more into MRI guidance into irradiation studies, and the icon fits in quite well, because literally the icon is in the same room as the irradiator, which I think was pointed out earlier. We don't usually put magnets besides X-ray machines, but because it's so innocuous, we're able to do that. And so now, of course, it's possible to see things on CT, but it's not nearly as clear, for example, as you could see, for example, in the MR. Now this was acquired with the M2, which is simply Aspect's version of the Bruker. Now Bruker sells them as the icon. And you can see very nicely visualization, and this is an ME180, or probably a prostate tumor model and a cervix model. We looked at 5 in the prostate and 10 and demonstrated the ability to visualize quite nicely on the MR. And of course, you can't quite see it on the cone-beam CT, which is expected because of the contrast differences. But the two being literally about 10 feet from each other, it's possible to position, transfer, and then achieve MR-based targeting for soft tissue structures of interest. And we've looked at things, for example, using no contrast. This is a very quick liver image on MR. You can see, you couldn't see anything on cone-beam CT. We've also looked at using some contrast agents to try to compensate for this, but of course it takes a long time for them to accumulate, and it may actually affect the radiation dose delivered. So it's nice to have the MR directly. One of the last applications that I was just going to touch on before I wrap up was a project that I had with a guy named Martin Wojtowicz, and he's really interested in the effect of radiation on neurons in the brain. So several years ago, about 5 years ago now, he and I worked together and finally published a paper in April 2011 where we were using a conventional linear accelerator to conform the radiation dose to just the brain of these rats. These are Sprague-Daly rats. He looks at their memory characteristics, but he also then looks at the rats after radiation and looks specifically at markers of neuroblasts and young neurons in the control, and then in the irradiated using something called a double Cortin DCX marker. And we look specifically at how the depletion of young neurons was impacted by the applied radiation dose. And it's quite striking. At doses at the level of half a gray or less, you had about half the number of neurons left. So it's quite, these doses are no different than about 10 CT scans, for example. It's quite a striking reduction. And so now he's repeating that, and we're working with him using the icon to image the rat brain, localize the hippocampus within the rat brain, and then fuse that with the radiation therapy imaging system to allow us to deliver conformal 1 millimeter volumes of dose to different parts of the hippocampus to start to probe where these cells are and where they're migrating from by using targeted, localized induction of the death of those neuronal progenitor cells. This just shows that fusion allowing us to target based on MR and the CT on the irradiator. So I'll summarize there. Oncological interventions will become more personalized, and this will challenge our preclinical methods. I'm really excited about the precision that you can bring to preclinical research with these kinds of tools. Preclinical imaging approaches can provide critical proof of principle data to support refinement of personalized cancer medicine. The results for Avastin in combination with radiation therapy are really intriguing on that model now. The development of a comprehensive suite of imaging intervention tools for cancer is now possible and becoming more accessible. And the methods for pipeline workflow analysis need to be developed. This is one of the biggest challenges. You get this data, but how do you actually start to draw critical analyses out of this? And I think hopefully we'll get something from Bruker in the future on that. So thank you for your attention.